Hey, welcome to Virology. So, if you're watching this, it means you weren't there for the first lecture, and that's okay. This will get you started. This is Biology 445. I'm Dr. Ben Newman. The only thing I know in the world, as far as you're concerned, is a bit about coronaviruses. Really, nothing else. All right, so let's go through some stuff. And this is how the class is going to run. And I'm hoping this will build some little coat hooks in your mind. And we can hang some ideas on those later. So let's try it. All right? All right. All right. First thing when people think of about it with a virus is how you feel. You're like, oh, I feel like I've got the flu. You may feel that way, but think about it. If you were to inhale an entire spoonful of cinnamon, like people did back, I don't know, 3, 4 years ago, that would make you sneeze and cough and everything else. But that's not a virus. Now, the flu virus can certainly make you sneeze, cold virus can make you sneeze, bunch of viruses can make you sneeze, but I also know people that look at the bright light, like the sun, when they want to sneeze, and that's okay. A lot of things can make you sneeze. How you feel is unrelated to what's inside of you. Viruses can make you feel worse, but they don't necessarily. So it's kind of tricky. So, no, there's no way to tell from the disease whether or not you have a particular virus. You would do a specific test if you're being careful and good at your job, I would say. So, yeah, disease is not virus. All right. See, this is easy, right? Now, it's much closer to the truth to say that a virus is its genome. So a genome here means a string of RNA or DNA, and that string is going to encode some genes. And so here we have the yellow string. So that's the genome of a virus. Now every virus has a genome inside of a little capsule of some sort, and that capsule is the virion. So when you're thinking of a picture of, I know what a virus looks like this thing up here, that's a virion. That's like trying to identify a person by the car they drive around in. And if it's the Wienermobile, maybe you're on to something. But if it's a white Toyota, everybody's got one of those. You can't tell. Or in Texas, if it's a truck, yeah. Ford truck, you got no chance. Yeah. Because that's literally all there are on the road. So you want to follow the genome. When the genome is inside the virion could be infectious. Just like when you're in your car, the car may start or it may not. It kind of depends on the car, and you usually can't tell by looking at it. But when that genome has been delivered inside of a cell, now the cell is infected, guaranteed. That's really the only way you know for sure that a cell is infected, is if the genome is in there. And then a bunch of secondary stuff happens. You start making virus genes from the genome, all this stuff. The genome of a virus isn't made of anything special. You can't use a magnet and pick those up. It's like a social security number. So my social security number, like this one of Mr. John O, it should be Q. I don't know, public. Mine has a 9 in it. I'll let you have that information for free. Yeah. I'm not going to tell you the rest though. Yours may have a 9 in it, and this one has a 9 in it. They're not the same number though. It's the same building blocks, which we call digits, in a different order, and it's the order that matters. Yeah, same thing with a virus genome. It's made of the same stuff you would find in a cell. Same ACGT or ACGU if it's RNA, but they're going to be in a different order, and it's the order that's important. So when people are saying, I think this virus came from there, I think that's how I can. No, you don't know that unless you are looking at the genome, and the genome's too small to see. So you have to do tests like we call it sequencing, which is how you read out the little parts of the genome. Or you can do PCR. That's how you copy different parts of the genome without knowing exactly what it is. And then you would sequence the genome, and that's how you know whether a virus is actually there. Everything else is what people who don't understand molecular biology think is the way you find a virus. Yeah. Which is fine. I mean, people don't know until they come to a class, and some of them don't even know after they come to a class. But this is how you learn. All right, let's go. Now, we're going to talk a lot about how viruses do the things that they do. We're going to talk a lot about enzymes. And an enzyme is just a kind of protein that does a job. Like you're a kind of person that's doing a job, and I'm a kind of person that's doing a job. Am I a virologist or am I a human? I don't know. Yeah, you decide. Yeah. But they're proteins, and these proteins fold up into a shape. The proteins actually move. You have to think of proteins as like bendy, stretchy, opening, twisting, closing, and doing work. They will do chemistry, they will move along physically. Everything that happens inside your cell happens because a protein is doing it. Yes, that's an oversimplification, but yes, it's pretty close to correct, so you can work with it. Yeah. There are proteins already inside the cell. Cell's like a big ball of proteins inside of a plasma membrane, and the proteins are all doing different things. Viruses grow inside of cells because viruses need a whole bunch of proteins that they can find inside of a cell. Viruses pack light. They don't have everything that they need, which is why we don't generally call them alive, but they're made of the same stuff and they require the same processes. They're life adjacent. Yeah, they are a walking, talking gray area if viruses could walk or talk, which they cannot. So when viruses go into cells, they make special virus proteins that are going to do virus jobs. Now, we're going to go through this course. We're going to start out hammering the heck out of HIV. We're going to learn everything about that because when we get to the next virus, we're going to be able to say, hey, it does the same thing HIV was doing, but instead of calling the protein one name, we're going to call it a slightly different name. And instead of working, this way, it's going to work this way, slightly different. We're going to have fine differences that you can hang on the hooks that HIV is going to be. It's not going to build them. Yeah. For every virus, you're going to be able to look for three things, and they aren't there in every virus, but they're there in enough viruses that it's worth building these mental hooks. There's going to be something to get the virus into the cell, to get the genome across the plasma membrane into the cell. And that's hard. Usually it involves physically punching a little tiny hole in the cell, but in a way that the cell does not burst and die. So, tricky stuff. Viruses are good at their job. Yeah, you're probably good at your job. That's fine. There's going to be a virus copying protein, and it's there to copy the virus genome. There are some viruses that are lazy. They're going to use the cell to make a copy of their genome. Yeah, those are disgusting viruses, honestly. But we'll talk about them anyway, even though I don't like them. Viruses, though, will most often carry a thing called a polymerase, which is just a genome copying protein. And viruses are going to carry a version of that works very well for copying the virus and not at all well for copying anything useful to the cell. Because viruses don't care about the cell. Viruses are here to copy themselves. Yeah. They don't care how you feel. They don't care how you feel after they're gone. Viruses are users. They're pushers. They're abusers. Viruses are terrible. Yeah. Don't ever, I get enthusiastic about viruses, but they're awful. They're the worst. Last thing, virus needs some kind of protection. It's going to need a packaging protein, a thing to make that little shell that goes around the virus genome, that thing we called the virion on the other slide, which is actually what you call it. And so there are going to be one or more proteins that are involved in protecting the package. All right. When you look at a virus, or rather a virion, you can sometimes spot these things right away. The parts that are going to grab onto a host cell, like your cell, and force away inside. It's usually the same protein. It's usually just one. And it's going to be all these yellow things. They're always going to stick out on the outside of the virion because where else could they be? They're like little Velcro hooks. They're going to catch on to your cells so that the virus can stick to the right cell, because going into the wrong cell is not going to get it anywhere. And viruses, they got to be efficient. A virion has basically one shot to shoot. It has one chance to get inside of a cell, and if it messes that up, it's dead. Yeah. And the genetic package inside is probably not going to go on to start another infection. You're sad, I'm sad, the virus is sad. Everybody's sad about that. The green things are going to be our protection. They can take many different forms. The protection parts are weird, but they're always there. And usually, because the virus genome is going to be RNA or DNA. Now, you remember what that stands for? Ribonucleic or deoxyribonucleic acid. If it's an acid, that means it has a negative charge. And so the proteins that are going to stick to it are going to have a positive charge. And you're going to learn which amino acids have a positive charge because there's only really two of them that commonly do. And yeah, sure enough, if you want to find the protein that sticks to the virus genome, it's got a whole lot of these positive amino acids in it. It's a positive little protein. Yeah, easy to spot. No big deal. Okay, you're going to look for stored energy. You're not going to follow the money. You're going to follow the free energy. And we're going to go from high stored energy, and we're going to dump that out step by step and get to lower stored energy. It's kind of like, when I say stored energy, it's kind of the way a Jack-in-the-Box stores energy. So there's a person at the beginning that actually punches that stupid little clown thing down into the box where it belongs and closes the lid, as it should be. Yeah, Jack-in-the-Boxes. Terrible toys. Yeah. Then there's going to be some other external energy. It's going to rotate this little thing all around the mulberry bush. And then you're going to throw a latch when you get to the right point of the song, and out springs the horrible little clown creature again. The spring was coiled using energy at the beginning, but then once it's coiled, it's storing that energy. And that's exactly how virus proteins work, just like a coiled spring. So here are some examples. We'll cover these in more detail later, but they are going to hook into, so the virus is at the bottom, the cell is at the top, they're going to hook into the cell. That's the first step, just like the little Mortal Kombat dude. Oh, get over here. Boom. Yeah. All right, don't look at that. That's violent. They're going to hook into the cell. They're going to reel the cell in. They're actually going to crash the virus membrane, if it has one, into the cell membrane. And that is going to make a little opening. And that little opening is the hole, because the virus genome is waiting right down here, and the inside of the cell is right up here. And once you crash those two things together, now the virus genome is inside the cell. And that cell is infected. Yeah, that's how it works. Bam, get over here. All right, yeah. Wrong way. All right. Another thing to look for inside of a virus in its genome is going to be a specific virus copying protein. Because while some viruses rely on the host for just about everything, most viruses are going to take charge of copying themselves so that they don't have to wait. Viruses don't want to wait in line, and so they'll grudgingly make a protein that just spews out tens of thousands of copies of the virus genome. We tend to call these virus polymerases. It's a particular type of an enzyme. It looks like a hand, and it's actually going to open and shut like a hand. There's a part called the fingers region, a part called the thumb region, and an active site, which is like down here. Like if you're going to grip an apple, that's how you do it. Yeah, so polymerases, like a hand. Yeah. What's the virus do? What's the virus doing? Like if you're looking from the outside, what would you see? This is the virus replication cycle. Why didn't we call it a life cycle? Because that would mean it's alive. And I'm not able to make that call. You know, that's way above my pay grade. Yeah, virus is alive? I don't know. You're going to have to decide that for yourself. Or just where to put it on that sliding scale. Because I think there's a sliding scale between alive and dead, or never alive. And honestly, it's theologically and biologically confusing. Psychologically, maybe more than either of those. So the virus is going to be on the outside, and it's going to be fully fit and ready, like this little thing up here. It is going to have all those coiled springs, and it's going to have that one chance to pounce on the cell, punch a hole in it, and get the genome out of the virus and into the cell. Before that happens, so we call this the inoculation stage, when the virus is bumping into the cell and finding it. Viruses don't swim. Viruses don't have propellers. They are carried along by air currents, like when you're breathing. You're pulling all that air in, and any viruses in that air are going to go bump, bump, bump all down your trachea and all through your little lungs until they bump into the right cell, and then boom, it's a virus bonanza. Then the virus is going to go quiet because stuff takes time. Yeah, let the virus cook, all right? The virus is going to go into the cell, and then it's got to unpack. Ugh, just like you on a vacation. It's got to decompress for a minute. It's got to start making proteins to copy itself. Once it's copied itself, it's got to copy all the parts that make up the spiky ball, the virion, that they're going to get it back out of the cell and into the next cell. And it's got to go around like switching off parts of the cell that would be annoying and like cry for help or scream in terror. Viruses can't be dealing with that sort of stuff. And so all of that takes an amount of time. For most human viruses, the minimum end is something like 5.5 hours to go from entering to coming back out, basically. Then we're going to have the burst, because a virus, one virus enters, and it's the opposite of the Thunderdome. Yeah, where one or two people enter and only one leaves. One virus enters, and you'll have 10,000 or more come out. The virus is going to turn that cell, which is full of DNA nucleotides, RNA nucleotides, little parts that can be assembled into proteins, and it's going to turn that into a virus factory. And now everything inside that cell is going to be used to produce viruses, and when it runs out of stuff to make viruses, it's just going to sag and collapse or burst open or die. It'll die. Yeah. Viruses use up cells. That's what they do. And all this time, viruses are coming out in ones and twos and tens and hundreds and thousands until you get this point where you've got all the viruses outside the cell, and congratulations, virus. You did it. Cell's sick or dead at this point, and the virus is on to the next thing. And it's got to do this every couple of hours, and that's what it is to be a virus. Then, when we get toward the end of the course, we're going to zoom way out. We're going to go big picture. We're going to look at what are some of the ideas about the origin of viruses? Because unless you have a time machine that can go back a couple billion years, I don't think you can actually tell me the origin of viruses. Yeah, any virus, really. But there are some pretty good clues that are left in the molecular biology. So we'll go through those. And then we'll talk about some of the really interesting stuff, things that aren't quite viruses. They're things called viroids, and they are super weird, but they kind of play by virus rules. And so who are we to keep them out of the club, right? Yeah, let them in. We're going to talk about mutations, which are almost always bad for the individual, but how having the right number of mutations can actually build a team of very defective versions of the virus that actually work better together than they would if they were a single, same number of copies but of exactly the same thing. It's kind of ridiculous. It's called quasi-species theory, but there's actually cooperation between these things that have no brain and no ability to think or coordinate or anything, and yet they do. It happens automatically. It's kind of neat. So, yeah, that's some of the stuff. I hope this gives you a little overview of where you can stick the different parts of information. All the class is going to have essay-based tests. And so you're going to fill your mind up and you're going to dump out all of that information in there. You're going to know the essay questions before you go into that because they're already up on the Canvas site. And there are only 10 of them or something. Yeah, for each test or fewer. Yeah, some of these tests only have 4 or 5 questions. You'll only have 2 questions on your test, but you usually won't know which 2. So you'll have to study a couple of questions. But yeah, honestly, it's not that bad. You're going to take these little bits of information, and instead of storing them as trivia, the little items, you're going to put them into connected stories. Because when you've got a story that has a beginning, a middle, all these things happen for a reason, you will know a whole lot more about molecular biology, and you'll kind of understand how cells work and viruses work and how biology works. That's the goal of this course, is to get you a point where you've got enough integrated knowledge that the world of biology actually makes sense. So we'll learn it through viruses because, I don't know, whatever. I like viruses. I'm enthusiastic about viruses. Could have done it with anything, but yeah, trust me, this is going to be good. All right, thanks. That's all.